Good morning, everyone. We welcome you and we thank you for your interest in this Conda Lab Talk. Today, we begin with a new series of webinars, the cosmetics testing. My name is Aletia Cruz, and I am a product manager of microbiology at Conda Lab. So, as some of you may know, Conda Lab Talks are webinars we prepare bi-monthly on topics of microbiological interest. And today, as you can imagine from the title, we will talk about the importance of cosmetic product samples treatment and also the recent ban on Triton X 100 and its effects on our analysis. We would like to mention that this platform, GoToWebinar, has a question box. So any questions can be sent to us this way, and we will take a few minutes at the end of the presentation to answer them. So we'll start with a quick overview on the cosmetic product and microbiological risks involved, followed by the importance of the preservatives and neutralizers in the analysis of these samples. We will continue with a general view of an analysis according to ISO regulations. Then we will get to the topic of the Triton X 100 and its prohibition, and finally a small perspective of current and future trends of the cosmetic industry, and to end, of course, the Q&A session. So let's begin with a bit of cosmetic microbiology and what is defined as a cosmetic. So a cosmetic is an article whose objective is to clean, beautify, or alter the appearance by rubbing, pouring, spraying, or any other application to the body. That is not only superficially, but also on teeth and mucous membranes. So by this definition, we find a great diversity of products commercially available that meet these broad objectives and that are composed of dozens to also maybe hundreds of ingredients. And because of their application, there are obviously concerns for the safety of these products, as microbiological contamination poses the greatest safety risk to consumers. So in this sense, cosmetics must be stable for a long time, taking into consideration that they will have a continuous use and that there may also be a prudent misuse that must also be considered. Microbiological safe cosmetics involve, as in any other industry, prevention through appropriate good manufacturing practices, and in the specific case of these products, and most importantly, normal and reasonably foreseeable conditions of consumer use. We can do a quick comparison with other industries, for example, the pharmaceutical industry, where unlike the cosmetic industry, they have highly controlled aseptic manufacturing systems, as well as a highly controlled form of application through prescriptions and the supervision of doctors. Another case is, for example, that the microbiological safety of foods are also supported by additional protective elements such as refrigeration, or even on the consumer senses, smell, sight mainly, that can alert the existence of deterioration. But we may think that the use of a contaminated cosmetic is not as serious as a contaminated food or medicine. And it is generally true that most infections through a contaminated cosmetic are transitory, superficial, and limited to where it was applied. However, there are much more serious cases, for example, when the contaminated cosmetic is exposed for example, to the damaged skin, and sometimes serious eye infections have been reported from the use of contaminated mascara, which have resulted in blindness. We also can find serious infections that can become fatal and are mainly associated with vulnerable groups, which would be the children, the elderly, also immunosuppressed, or chronic diseases such as cancer, diabetes, AIDS, can make an apparent harmless infection a death threat. Also, we could have temporary conditions that can affect this, such as pregnancy or previous existing infections and damaged skin. So, what are the main microbiological risks that can contaminate a cosmetic? We have to begin with the composition of this product because it contains a source of nutrients that allow the growth of microorganisms, and this would be a carbon, nitrogen source. They also can have growth factors such as vitamins, trace elements that are components of minerals such as clays or talcs. Another issue is the risk determination factors, and for this, it is necessary to evaluate certain characteristics of the product, such as water activity, which is one available in the formulation for the microorganism, and it is an effective way of preserving the product when you restrict the available water. Some products can be creams and balms. They have very low risk, very low water activity, so the risk of the contamination is also low. Surface tension and oxygen distribution, pH. Some products such as chemical exfoliants have a very low pH below 3, or some straighteners, hair straighteners have a pH above 10. So the development of microorganisms would be quite difficult. And also the presence of inhibitors such as alcohol or raw inhibitory materials such as oxidizing and reducing agents that we can find sometimes in deodorants and antiperspirants in some products like that. So we can also look at production because it should be considered, since if it was carried out at a low temperature, it would slow down growth, and conversely, a production at a high temperature, higher than 65 degrees Celsius, would mean the thermal death of the microorganisms. So in fact, if the filling temperature is higher than 65 degrees Celsius, the microbiological analysis should not be carried out. And also the container, the packaging. We must consider that it is an additional protection, but its level of safety may differ. We have, if it's a single or a multi-use container, the size can also affect the way of dispensing, the expected duration of use of the product, and also, and more importantly, if there's any direct contact with the customer and the product, and also the pressurization. So the type of packaging used, package use will influence the concentration and activity of the preservative, and the containers obviously should be tested to determine the effectiveness of the preservative under actual storage conditions. So we have that according to ISO 29621, some low risk products identified due to the compliance and combination of the above would be powders, loose or compact, lipsticks, toners that have alcohol in the formulation, antiperspirants, and soap bars. And on the other hand, as a high microbiological risk, we would have emulsions, alcohol-free toners, and some gel preparations, which would, these high microbiological risk products would require the addition of a preservative and also microbiological control. So as we can see in this table, the potential microbiological risks are numerous, including both bacteria, gram-negative, gram-positive, and fungi. Some are derived from manufacturing and others from consumer use. However, the range of microorganisms can coincide. Burkholderia and Pseudomonas are the most common production contaminants, and the contaminants by consumers, because of the contact with the product, are usually opportunistic microorganisms which are found on our skin, such as Staphylococcus, Streptococcus, and Corynebacterium. Some that we can assume are exclusive pollutants from production because they are attributed to raw materials would be Clostridium, which can be found in talc, for example, which is derived from the soil. And during production, we can find contamination from raw materials, processed water, poorly designed equipment, ineffective cleaning and disinfection processes, of course, poor hygiene of workers and facilities, and lack of training and awareness. And furthermore, consumer contamination is very diverse, given the various microbiological exposures and practices all around the world. However, for any type of contamination, gram-negative bacteria pose the greatest risk due to their widespread presence in aqueous environments and their tendency to adapt and grow in products, and very importantly, to develop resistance to preservatives. So let us now go on to review the need for preservatives in cosmetics and their subsequent neutralization for a correct microbiological analysis. The objective of the preservation of the cosmetic product, which extends, as we've seen, from the quality of the raw material to the safety of the last use of the product, is to establish and maintain the microbiological quality to ensure that the consumers are exposed to a minimum microbiological risk, always considering reasonable foreseeable use, which is very important. What are the consequences of not meeting this goal? We could have loss of product aesthetics and functionality. Obviously, consumer safety would be compromised, and both fungal and bacterial contamination can physically compromise a product with sediment production and emulsion breakage. But unfortunately, most often microbial contamination has no discernible effect, which denies the consumer a signal that the product being used may pose a potential safety risk, unlike, for example, food, which is very obvious sometimes when it has gone bad. And as we've reviewed above, the preserving of a cosmetic is achieved by controlling the risks associated with the non-sterile manufacturing, good manufacturing practices, and the risks of contamination of the consumer associated with reasonable use. However, there are products that, due to their packaging, are not subject to user contamination, so the sole purpose of a chemical preservative is focused on contamination during manufacturing. The efficacy of preservatives should be tested by a preservation efficacy test, or better known as the challenge test, which we will get to in a moment. And since preservatives are chemicals after all, even if they are used to protect consumers, they also have the potential to cause harm. Therefore, its regulation is a matter of vital importance. Both the European Union and Japan have developed lists of allowed preservatives and have also prohibited the use of others. In the European Union, cosmetics are regulated by the Regulation 1223 of 2009, which includes an annex with pre-approved preservatives that can be used in cosmetics. We can also find within the microbiological specifications for each ingredient used in cosmetics. And this regulation defines preservatives as a substance that can be added to cosmetic products with the main purpose or objective to inhibit the development of microorganisms. And some requirements to consider for a preservative are their microbiological activity, to reduce the number of contaminating products to a very low concentration, microbiostatic activity to inhibit the growth of surviving microorganisms, control recontamination during the habitual use of a consumer, safe, that it is safe for the consumer, which this is kind of achieved by having a minimum concentration of the preservative in the product, broad spectrum of microbial action, that it does not modify the organoleptic characteristics of the cosmetic, that it has chemical stability and compatibility with other components of the cosmetic formulation, and that it has a high ratio cost effectiveness. So as well as their interactions with the cosmetic formula, for example, an alteration in the pH can change the ionic character of the preservative. Some preservatives combine to surfactants that in some cases can impede antimicrobial activity. Some proteins found in the cosmetic formulation can inactivate preservatives. The emulsions with all bases can redistribute the preservative and change the concentration level in the aqueous phase. Also, the buffer system of some formulations can affect the activity of the preservative and inhibit them. Some solids can absorb preservatives, so a higher concentration would be required. So certainly the elements of the manufacturing processes, the formulation, the packaging can contribute both to the effectiveness of the preservative and to compromise it, as well as its stability. To measure this effectiveness, ISO 11930 provides us with a method to evaluate the conservation of a cosmetic, which is applied to any commercialized product, with the following exceptions. Products in which a low microbiological risk has been demonstrated, and in products of single use, or that the packaging does not allow a direct contact with the consumer. The challenge test, or the preservative efficacy test, determines the effectiveness of the preservatives present in a certain cosmetic formulation against certain pathogenic and or altering microorganisms during normal use by the consumer. It is carried out when there is development of new products. Also, if, for example, the formula or even the manufacturing conditions of our product already commercially available are modified. The test consists of inoculating the cosmetic with standardized inocula separately for each strain, of course. So to each sample of cosmetic, you add the standardized inoculum and mix thoroughly to obtain a homogeneous distribution of this inoculum. The number of surviving microorganisms is measured at defined time intervals each 7 days for 28 days. So you take a sample and make decimal dissolutions with a neutralizer, and then use plates used for counting, TSA for bacteria, SDA for candida, and PDA for aspergillus. So finally, the calculations are made. So and also the log reduction values, that is the logarithm of the number of microorganisms inoculated in time minus the logarithm of the number of surviving microorganisms at each sample time every 7 days. So these values are compared with the minimum values required by the evaluation criteria, which would represent the protective capacity of the cosmetic formula. And as in any other final product quality control analysis, for cosmetic samples, we must also use a medium to dilute or enrich the sample to be analyzed. However, the preservatives present in the formulation can cause problems in our results, since, as we've seen before, they would prevent the development of the microorganisms present, and thus we would obtain false negatives. That is why the use of neutralizers in these diluents or enrichment media is essential, so that they inactivate a preservative or biocide and at the same time allow the free growth of microorganisms. If a biocide or preservative is not inactivated, we can easily overestimate its antimicrobial capacity, because its activity would continue even during dilution or enrichment. Controls to measure toxicity to the microorganisms of the neutralizer should also be included during its evaluation, which can be determined by comparing growth in neutralizing medium versus growth in general mediums which would be TSA or SDA. So now we have a couple of types of neutralization. The first that obviously comes to mind is, of course, chemical neutralizers. So lecithin, polysorbate 80, sodium thiosulfate, and are some of the most common. They inactivate quaternary ammonium compounds, or QACs, phenols, and halogens. The neutralizer and the effective concentration must be determined for each preservative and microorganisms. Some biocides are difficult to inactivate. For example, the sodium bisulfite is the neutralizer for formaldehydes and glutaraldehydes, but then the sodium bisulfite also inhibits the growth of bacteria and spore germination. So in addition to its effectiveness, it is also important that the biocide is not toxic to microorganisms. So in this table on the right, we have the neutralizing agents and the preservatives or biocides that they affect. One of the most used and effective neutralizing media is the Day-Angley medium, which contains sodium thioglycolate, sodium thiosulfate, sodium bisulfite, lecithin, and polysorbate as neutralizing agents. However, some preservatives are sensitive to the concentration in which they are found, so they can effectively be neutralized by dilution. This has been reviewed in several publications, and one method recommended is the membrane filtration. So for this, bacteria are isolated on the filter and then washed free of preservatives. The surviving bacteria are then transferred to the culture medium. It has been suggested to wash the membrane with nutrient broth and then transfer it to the surface of a plate to count the surviving colonies. So let us now quickly review the ISO methods for a cosmetic analysis. So in general, the ISO method for cosmetic analysis indicates, regardless of the target microorganism, dilution and neutralization of the sample must be carried out. And it's very important, as we have already seen, for which the same enrichment broth can facilitate the blocking of the preservative. So followed by incubation and then isolation on a selective medium for each microorganism. And finally, the confirmation of the colonies that can grow is carried out. Also, the ISO indicates the suitability of the detection method. For these, plates are seated in TSA or SEA, which are non-selective, non-neutralizing media, to prepare the inoculum. The culture is collected and suspended in diluent to obtain an standardized suspension. Then serial dilutions are made with the initial sample suspension and mixed with the standardized suspension. It is inoculated in the selective medium, and as a control, we have an uninoculated plate. The detection method is successful when there is a characteristic growth of the organism on the inoculated plate, and also if there's no growth on the control plate, which is the uninoculated plate. And on the other hand, if we find growth on the control plate, it means that the product was contaminated. So here we can see the microbiological limits set by ISO 17516 that there may be presence of total mesophilic aerobes, but not of pathogens such as E. coli, Pseudomonas, Staphylococcus, or Candida in 1 gram or milliliter of product. So the media used in cosmetic microbiological analysis according to ISO vary in the selective medium depending, of course, on the microorganism to be analyzed, but we can find practically the same enrichment broths. So there are 3 mainly used, which would be the Ogon LT100 broth, Day-Angley neutralizing broth, which we mentioned earlier, and the lecithin modified broth. Others that can be used only when there are no preservatives found or present in the product are the maximum recovery diluent, buffered peptone water, and lactose broth. In addition, we have another medium, the Behrens diluent, which is not an ISO medium. However, it is used routinely in the industry as it complies with the recommendations, including the most common neutralizers such as lecithin, polysorbate, and sodium thiosulfate. And why is it so important to know about preservatives and neutralizers for isomedia for cosmetic analysis? Well, because sometimes these substances can undergo changes in their regulation. To have some context on the regulation of substances in the European Union, the European Chemicals Agency, or better known as ECHA, is the organism that is responsible for regulating substances in the European Union. And they have become the regulatory authority with the largest database in the world, currently containing more than 245,000 substances. And one of its objectives, in addition to protecting human health, is to reduce the environmental impact of chemical substances. For this, the ECHA, or ECHA, has established more than a decade ago a regulation called REACH, whose initials refer to the registration, evaluation, authorization, and restriction of chemical substances. It must be remembered that the microbiological quality control of cosmetics is carried out in order to protect consumers. And in this sense, REACH protects the health and the environment against risks derived from chemical substances and mixtures, which applies to all chemical substances, not only those used in the industrial aspect, but also those used in the everyday life. However, REACH places the burden of these tests on companies that must comply, and it's their responsibility to demonstrate to the agency the safe use of a substance, as well as the communication of the associated risk management. If the risk cannot be managed, then the authorities, the ECHA, can restrict or even prohibit the use of a substance in question. And then why are we particularly interested in ECHA or REACH? So in 2012, the group for ethoxylated phenol, which includes the Triton X 100, was on the authorization list of candidate substances as extremely worrying due to its endocrine disrupting properties in the environment. And 5 years later, it was included in the Annex 14 of REACH, the octylphenol and nonylphenol ethoxylates, indicating the substances requiring authorization for use, which also includes the Triton X 100. In other words, its generally use in the European Union has been banned. And January 2021 was the sunset date for which these OPE and MPE substances cannot be used unless an authorization is granted by the authorities or the use is exempt from authorization, which is very sensitive and usually reserved for very high profile activities. And for this reason, it has been recommended to promote the evaluation and use of alternative substances that are friendly to the environment. The importance of this decision by ECHA lies in, again, Triton X 100, which is a detergent that is included in the composition of some culture media for the cosmetic industry according to ISO standards. It is an excellent dispersant that improves the diffusion of microorganisms, and the affected media containing this substance would be mainly the Eugon LT100 broth and agar. So that is why from Condalab we have alerted our clients about this situation and offered an alternative media to continue carrying out the quality control analysis of cosmetics according to ISO. For this, we have the Dayangli broth, which, as we've mentioned previously, it is an excellent alternative. We also have the lecithin modified broth, lactose broth, and the Ringer solution. Another possibility would be to perform the analysis under pharmacopoeia. However, the change would be a little complicated due to the need to perform the validation required for the implementation of a new method. A good media could also continue to be used, but without the Triton X100. So it would not be really, you would not be really following an ISO method. And well, the simplest thing would be just to use one of the alternative media that it's available. So to conclude quickly, we will comment a bit about the current situation of the cosmetic industry and what we can expect in the future. So it's obvious the interest and demand of consumers for organic products, which has been around a couple of years and increasing lately. This has generated a rejection of traditional lifelong preservation systems, which we can easily see replicated in other industries like the food industry mainly. The substances that we have heard the most are also marketing strategies such as paraben-free or formaldehyde-free. And that is why consumers are now opting for natural preservatives, which have led to an application to satisfy the consumer, but without really understanding the accompanying contamination risks. And in recent years, this lack of understanding of the risk involved has even led to compromising the product quality, which is very worrying. There's also the tendency of using processes that minimize the microbiological risk that we mentioned earlier, which would be the filling temperatures either or the production temperatures, either high or low. Also, a good packaging that does not allow contact with direct contact with the consumer, etc. And on the other hand, we have that in the European Union, there are also almost 60 preservatives that can be used in cosmetic products. However, we only see a handful of these in most of our products available, which means that there is a greater exposure, and thus the incidence of associated health problems increases, and it creates a mistrust about the safety of the rest of the authorized preservatives. So we should really start using the other ones to prove the consumers that if they have been authorized by the ECHA, they are obviously they pose no they pose no risk to the consumer. And like in any sector, the cosmetic industry must also find ways to reinvent itself to meet the demands of its customers, of course, without compromising the risk. And most importantly, always remember that there is no perfect preservative, but the most suitable for each type of formulation and consumer. So at Condalab, we have an available complete catalog of products according to ISO in different dehydrated or ready-to-use formats for cosmetic analysis. Of course, our products have controls throughout the production processes, from the raw material to the final product, and each batch produced also has its own microbiological control. We are, as you can see, aware of any changes in the regulations or legislations to always offer products with current formulations and, in this case, neutralizers. So with this slide, we conclude the webinar. We will take just a couple of minutes to answer some questions that you have been sending to us. So, okay. How much product sample is required for the challenge test? So as we mentioned, the challenge test is by following the ISO 21148, marks it to be at least, I think, 20 grams or 20 milliliters to prepare the initial suspension. So you can consult the ISO, but it should be that. Okay, so we have a couple more. Does the neutralizer necessarily have to be added to the dilution broth? So most procedures it is require the chemical neutralizers to be included in the enrichment media or diluent. As you've seen, the ISO media includes in their formulations the neutralizers, but depending on your process, you can also just add it in the culture plate. So there's that, but the ISO media already includes the neutralizer. Okay, acceptance criteria for pathogens and microbial load. We have already covered that. We had a small table with the, we can go back if you want, just quickly. Here, the microbiological limits according to ISO. This would be the accepting criteria, which would be basically absence for pathogens in 1 gram or milliliter. And then, well, we have here for the total mesophilic aerobes. Okay, so, okay, what are you contemplating alternative methods in cosmetic analysis? So currently there are no third-party accredited methods, but there are chromogenic media to have a rapid identification. Here at Candalab, we have been developing specific media for cosmetics for quite a few years. We have the Pseudomonas, Staphylococcus, but we will launch some new ones in the coming months. An alternative method that allows a percentive identification. So please expect that as it's coming in a few months. Oh, ISO update to accommodate the ban, the Triton ban. I understand. So yeah, it's true that the ISO should be updated, and well, at the moment we have no information about it. We are in contact with the organization and certifying agencies, but it is complicated as it is a specific issue of a market. The European Union is not something global, so we don't know when there will be, and if there will be an ISO modification. Let's also remember we have alternative ISO media to use, so it's not as a pressing matter as, well, we think it's not a very pressing matter. So at the moment you do not supply Ogon LT100, or you supply it with a different formulation without Triton. Yeah, we have been doing some work in the lab. We have been doing some tests. We cannot, as it has been banned, we cannot sell, supply, or commercialize, or even use ourselves the Ogon LT100 with Triton. So we cannot absolutely do that. We are working on giving you, we're also supplying you with an Ogon broth agar with an alternative Triton substance. So we should have that ready in a couple of, I don't know, weeks maybe. We will let you know. So that's very, it's very important that you are, that you sign up for the newsletter, or that you follow us on LinkedIn, so that you can have all this information at the same moment we have it, so you are informed. We have a couple more of questions, but I don't want to take more of your time. So we hope that you have found this Candalab talk interesting, informative. If you require a certificate of participation or have any further doubt, please, you can reach us at micro@Candalab. We want to invite you to join us for our next webinar on Yersinia. And as I've said, don't forget to follow us on our LinkedIn, sign up for the newsletter. We send very important information through those channels, and we also send the dates and the webinars and the invitations, and that's it. We hope to see you in the next. Thank you.